Okay, good afternoon, everyone, and welcome to today's webinar. Thank you all for joining us. We appreciate you taking the time to be here. My name is Gerard Mangan. I'm an application scientist at Quantum Detectors. I'll be hosting today's session where we'll hear from our speaker, Helen Leung, on 3D ED and 4D STEM as complementary acquisition modalities. Before we begin, just a few quick notes. This webinar is being recorded so that you can revisit it later, and also for the benefit of those who couldn't make it today. If you have any questions during the presentation, please type in the chat. At the end, we'll have a dedicated Q&A session for 5 to 10 minutes, where I'll read out your questions for Helen to answer. So let me introduce our speaker. Helen is a PhD candidate working with Professor Paul Midgley in the Department of Materials Science and Metallurgy at the University of Cambridge, alongside her industrial sponsors GSK. She works on the application of multimodal electron and X-ray diffraction techniques to materials of pharmaceutical interest. She holds a master's in material science degree with first-class honors from the University of Oxford, where in her final year, she worked on developing convolutional neural networks for image analysis in electron micrographs. Helen is interested in the role that characterization techniques will play in the development of new nano-engineered drug modalities and how these technologies will support the era of precision medicine. So with that, I'll hand it over to Helen.
Brilliant. Thank you very much, Billy, for the introduction, and thank you everybody for joining today. I will aim to speak for about 30 minutes in total, and we'll be speaking about a couple of projects that I've been working on essentially throughout my time at Cambridge. And please feel free to put any questions and ask any questions in the chat for afterwards. So firstly, a bit of an introduction as to the sorts of work that I'm doing. You'll notice I work with industrial sponsors GSK, who are a large pharmaceuticals company. And the idea of the research is to understand how electron diffraction, specifically as a characterization technique, could potentially be helpful for the pharmaceutical industry, particularly in drug development. Obviously, there's been lots of interest in electron microscopy in drug discovery, so up in the proteins end of the world. But that's not what I'll be talking about today. I'll be talking about small molecules and the drug development, so further downstream in that. So I'll first introduce the two techniques that I'm talking about and just give a very brief rundown of what they are and how they work in the microscope for those of you who aren't familiar with either one of them. And then I will present two case studies, one on a system called indomethacin, which is a drug for arthritis and for gout, for example, and then a second case study on xanthine, which is another organic molecule of interest. Okay, so 3D electron diffraction, some of you might know it as micro-ED, it's also got many other names in the literature, is analogous to single crystal X-ray diffraction, and it's essentially where we take a parallel electron beam in our microscope, so we're in TEM mode, and we tilt a crystal around a range of rotation angles, and at each rotation angle we would collect one pattern, and this gives us essentially a tilt series. And when we reconstruct that in reciprocal space, we end up with a data set that's essentially analogous to a single crystal XRD, so classic crystallography, and we can therefore extract atomic positions but also molecular conformations. And the reason why this is particularly interesting is the size of the crystals that you can have access to, which are micron-sized. And this is because in the electron microscope, we can focus an electron beam down into an illuminating area of maybe 1 micron or slightly larger. But so we're able now to access much smaller crystals. And so this is great because if you have very scarce samples, you don't have a lot of the sample, you are able to still do a structured solution potentially. Or if your samples are not able to grow very easily into crystals large enough that you would send to the synchrotron, for example, or just to do single crystal XRD on, and this technique is quite a good way to do it with comparatively minimal sample prep. So just a quick workflow for those of you who are unfamiliar. This is just quite a standard crystallography workflow. So you might prepare your sample in the 3D ED case. we're not having to make any particular effort to grow our crystals. So often we might just disperse the powder onto our carbon grids. We put that into the microscope, we acquire our data. There's many different types of ways that you could acquire that data. For example, you can process the beam, you can have a serial approach. But the goal is to collect your tilt series, which fairly represents, all of that samples, all of reciprocal space, or as much as possible of it. And you, from there, can do your peak searching, intensity extraction, understand what the unit cell and the space group is of your compound. And from there, work to recover the phase via structure solution and then a structure refinement. And so this workflow is pretty standard, and you can use, and if basically all the electron diffraction data can feed back into X-ray diffraction pipelines relatively easily, although this has been done quite widely, and so I guess there are differences between electrons and X-rays, and we can get onto that later. But broadly speaking, the workflow is very similar. Okay, so 4D STEM, so scanning transmission electron microscopy, is slightly different to the 3D EDU side, because this time we have a focused beam rather than a parallel beam. And so we're working with a convergence angle. In our case, we work with convergence angles of maybe 0.1 milliradians. And the idea is you scan your beam across your sample, and at each real space position, you collect one reciprocal space, so one diffraction pattern. And so this gives you this 4D data set of now spatially related diffraction patterns. So with 3D ED, you had rotationally related diffraction patterns. With 4D STEM, you have spatially related. But you end up collecting far more diffraction patterns than you would with 3D ED because of your probe size, which is much smaller. And so typically, in the settings that we use, we collect up to maybe, for example, 65,000 diffraction patterns per scan of an area, which will take about 1 minute for us. And that covers an area on the order of microns. Why is 4D STEM important? Well, aside from understanding the crystal structure, understanding the crystal's microstructure and the microstructure of product is also very important. So the crystal structure might influence the way that a crystal is expressed. So for example, its habit and its morphology. And it would also therefore potentially impact the microstructure. And that goes on to influence the macrostructure. And the material scientists among us will know that link between the length scales is very important. And so on the pharmaceutical scale, understanding, for example, how your API, so that's an active pharmaceutical ingredient to a drug, might interact with an excipient, is really important to know how these things can be manufactured and scaled up. Okay, so going on to our first application of indomethacin. So amorphous solid dispersions are a formulation type, and they are used for any sort of drug which has very poor aqueous solubility. And these drugs are going to be normally ingested, so you would take them orally. And of course, if they've got poor solubility, by the time they get to your gastrointestinal tract, the passing of the drug basically through into your blood is dependent on that solubility somewhat. So if we have a drug with poor solubility, what we might do is we might wrap it into a solid dispersion and fill it with excipient, which is a polymer. In this case, we used PVP. And ideally, you get in what we have in section number 1 here, which I have labeled homogeneous, which is that you have this very evenly dispersed amorphous mixture, which has superior solubility properties. But over time, because this is metastable, you can get phase separation of this polymer in your drug. And eventually you can get unwanted crystallization, which can affect the shelf life of the drug and also the performance potentially of that drug. So we were interested to see, what does the crystallization in an amorphous solid dispersion look like? And can we see that with electron diffraction? And can we understand potentially the origins of the crystallinity and the whereabouts? So the crystallinity can take many forms, for example. So in indomethacin, this drug, and there have been a handful of great 3D ED studies on this as well. There are different crystal forms, which we call polymorphs, and those different polymorphs might have different stabilities, different bioavailabilities, and importantly, different toxicities. And for those of you who are interested, probably the most famous example of where polymorphism in a drug has gone wrong is in ritonavir, where, yeah, essentially at the late stages of that drug, an unexpected polymorphic form was found in the manufactured product, and this had non-ideal effects. So we applied 3D electron diffraction firstly to our amorphous solid dispersion, looking at crystals within the powder, within this formulation, to see what crystal structures we had. And we found a new form of indomethacin, which the unit cell didn't match essentially any of the unit cells that had been seen before. And this was done at cryogenic temperatures on a cryos, essentially. And we can see the crystal there, so you can see the size of the crystal we collected from, and you can see the tilt series as well. And this essentially was able to give us a structure solution, which I say trivially, but it actually took a long time to do. And what's really important about this is the way that this indomethacin molecule is molecularly, in terms of its conformation, can tell us a little bit about the way that it came about. And so we call this this new polymorph the sigma polymorph, and you can see that it matches up in terms of conformation quite well with itself. But one of the things, I'll come into here, so we can start comparing that to itself and understanding the conformation of that molecule. But also, I want to point out C in this slide, which is these yellow circles that you might see on the slide. These represent channels that we could see. So we noticed that there was quite a lot of space in the structure. And what we realized, or the hypothesis from this direct insight from the structure, was that there had been a solvent in those channels, which was helping to kind of template the crystallization of this specific form. And that solvent was no longer there. And you can imagine, with large channels in your crystal structure and a relatively volatile solvent, that is evaporating quite easily. And so what we were able to do is we were able to remake our polymorph and reproduce this polymorph just using the solvent now, and taking away any PVP, so any of this amorphous polymer. And we were able to prove to ourselves that this was the mechanism for crystallization. So I guess this is just one example of where the direct insights from the structure has led to a real life benefit in the manufacturing techniques. Okay, and we then applied some 4D STEM to these crystals. What was difficult before is, if you can imagine, we didn't know the crystal structure before. We didn't know the form of our polymorph before. So just applying 4D STEM to it without a CIF file, without any understanding of what unit cell parameters we were looking at, was very difficult, essentially. And so I don't have time to go into this here, but there is a bit of work that you can follow the link to on the bottom of this slide if you want, and read a bit more about when we applied basically 4D STEM to this now. But bearing in mind, we had to first successfully achieve structure solution through 3D ED before this was kind of a meaningful experiment to do. And what the slide is showing here, and the contrast that you can see in the virtual dark field images, is potentially essentially some twisting, but it's a really interesting example of the crystal microstructure and just being able to unlock that. But the key here is that's not really an insight that we can have without the 3D ED. Okay. Oh, and I should say that the indomethacin work is also published as well. So if you want to read up more about it, you can find that separately. So now I move on to xanthine work. The xanthine work that we've done is in two parts. So I will introduce the first part first. So xanthine is a purine base, and it's part of this family of molecules. For those of you who might have worked with guanine before, you would recognize that this is a very similar looking molecule. It's also relatively similar to other plain organic molecules such as caffeine, adenine, hypoxanthine, and we had some difficulty essentially. Well, guanine and adenine, the crystal structures of those are known, and those are bases in DNA. So those are quite well characterized and found in biogenic crystals. But xanthine, at the time when we were looking at it, there was no clear crystal structure for it. So we thought it would be quite an interesting case study to work on and to have a look at what electron diffraction can tell us about this system. So we dispersed our xanthine crystals, microcrystals, synthetic xanthine that we just purchased from Sigma Aldrich onto carbon-coated TEM grids. And you can see on the figure on the right in panel A, that dispersion is sort of what we were aiming for, because that's just kind of the ideal separation between microcrystals, which means that you can get a full rotational range without potentially other crystals coming into view, and they're nicely separated. So we ended up collecting a successful data set from xanthine, from a crystal which is, I mean, under a micron in size. You can see here on the panel on the left, I would estimate maybe 300 nanometers. There's some amorphous contamination on top of it, so you have to look quite carefully at the panel, but you can see underneath that there is a much clearer, darker shape underneath, which is our crystal. And we also collected this data in the same conditions as with the indomethacin. And from this, we achieved a structure solution. So xanthine, it being itself a planar organic molecule, the crystal structure it forms is also very planar and in layers. So the layers here you can see in panel A, they are stacked on top of each other. And this form that we found in abundance, we called it form 1, and it's a monoclinic form, essentially. What was very cool about our data is xanthine has tautomers. So the position of the hydrogen can flip between the 2 nitrogen positions on the molecule that you see here. And our 3D ED data was of high enough quality that we could distinguish the tautomers and confirm basically that we had the 7H tautomer, which just means we could see where that hydrogen was very clearly. And the resolution of our data, I think we cut to 0.9 angstroms and gave us an R factor of about 10.9%. So that was very promising and very cool to see that we could see that. And so this has been written up into a paper, which you can find in Crystal Growth and Design, and talked about basically the form 1 form of xanthine. Okay, so this is just 3D electron diffraction though. And what I want to remind everyone is that we're illuminating still a large area compared to 4D STEM and 3D ED because we have this parallel electron beam. And one of the things we noticed whilst we were collecting 3D ED data from xanthine was that some particles did not just have one reciprocal space lattice orientation. And it appeared for lots of our particles that there was some interlocking essentially going on between 2 reciprocal lattices. And so we suspected that this might be some form of planar disorder, or, well, twinning here specifically. But looking at the structure of the xanthine as well, you could imagine we've got very tightly bonded hydrogen bonded layers, and they're held together by relatively weaker van der Waals forces. And so you could imagine that there is some sort of flexibility that the structure has around the positions of these layers with respect to each other. So the first thing we did with the 3D ED data is we can calculate the orientation matrices of the 2 reciprocal lattices that we do see and relate them to each other. And so we found that we essentially had twins in xanthine where the composition plane, so the composition plane and twinning notation, I guess, is the plane that doesn't change, was the plane of the hydrogen bonded layers. So the layers in that you can see clearly of the molecules, those were not changing. So we were not breaking bonding within that. But by rotating or reflecting about the 101 direction, which is the direction you can see pointed out in C, that is basically our twin boundary. So rotating across that can essentially twins the xanthine crystal, keeping those hydrogen planes, which are the 10 bar 1 planes in this example, consistent with each other. So that was really interesting to see. So we thought, okay, maybe this explains a lot of what we're seeing in 3D ED. Maybe that will make sense. However, when we took this to X-ray powder diffraction, because of course we're working from a powder sample, so we can also take some of that sample and put it. And we went to the diamond light source, and we went to the beamline I11, which is high res XRPD. And we unfortunately, just with the Form 1 model, we struggled to really model this data very well. You can see that in the little blow up that I've got here on the slide, there's an extra peak that we couldn't really account for. The peak shapes were asymmetrical. They all seem to have some sort of right sloping shoulder. Just purely having the CIF file, well, the CIF for Form 1 xanthine didn't seem to be able to account for everything that we were seeing in the XRPD. And the presence of twinning as well wouldn't be able to explain this. So we went back to 3D data and thought maybe there is something more that is hidden within this data that is telling us about this behavior that we can see on the bulk. So we ended up finding another polymorph of xanthine, which is very exciting. And you can see the sections from it here. I'm going to now show you what the crystal structures look like. So A and B on the top right panel, that is of Form 1 xanthine, and C and D is of what we call Form 2 xanthine. And the first things you'll notice is Form 2 is orthorhombic and Form 1 is monoclinic. And these are their unit cells. But what we wanted to understand is how they're related to each other, since they seem to be so interchangeable. And actually what was very interesting is the structure solutions of the orthorhombic Form 2 structure came from a bicrystal where we could see two reciprocal lattices in that space, and we had to separate those reciprocal lattices and then run them both through structure solutions. And it was revealed that one of the reciprocal lattices corresponded to Form 1 xanthine, and the other one corresponded to Form 2 xanthine. So in other words, both Forms 1 and 2 were present in the same xanthine particle, which was like a bicrystal. And the way that they're related is, if you look at panels B and D on the top right corner of this slide, you can see that the layers are the same essentially. So the xanthine molecule arrangements within those hydrogen bonded layers is unchanged, essentially, their coordination. And so this makes this a very specific type of polymorphism, which we call polytypism. So these are polytypes of each other, and that's just a subset of polymorphs. And you can see where the extra reflections come in blue. I've highlighted in the sections here in panel B, which show where the extra reflections are introduced when you have orthorhombic symmetry. Okay, so we tried to model this a bit more by doing basically trying to understand what this layer interaction looks like and how these layers are related to each other. So on the left, I've labeled all the layers of xanthine Form 1, 1, 2, 3, 4, and then I've labeled the layers of xanthine Form 2, 1, 2, 3, 4, 5. Now each, just because of the way that the unit cells are defined, they each have 4 layers in there, like in one unit cell, but you can't see the 5th layer basically in Form 1 xanthine in this particular schematic. But we essentially labeled 5 layers from Form 1 and 5 layers from Form 2. And the goal was to understand how layer 1 was related to layer 2 in Form 1 versus in Form 2. So what we did is we took this is an example for Form 1 to show how I got to the figure from the previous slide. We took layers 1 and 2, and we wanted to understand what is the translation vector that I have to do to overlay layer 1 on top of layer 2. Obviously, we know that there is a depth translation, but we know that the distance between these hydrogen bonded planes remains relatively invariant. So what does the translation between the planes this way look like? And so here we thought about, how do I move the pink molecules onto the blue molecules? And so you can see I've shifted and done a direct translation of the pink molecules. I'll just play that one more time. The pink molecules onto the blue molecules here. And that's how I move from layer 1 to layer 2. And we call this translation A. And we repeated this with all of the layers for both forms of our xanthine polymorphs, of our polytypes. And the results are essentially here. So in form 1, you have a stacking sequence, essentially, which we call AB, AB, and that repeats, and that goes back to A. So it goes AB, 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 AB. But in form 2, in our orthorhombic polytype, we have AB, and then CD. So we have 2 new translations that are introduced in the orthorhombic type. And so now this goes ABCD, ABCD, ABCD. And so now we have different stacking sequences that relate the 2 polytypes to each other. Okay, so I've shown you in the panel up here on the top left what A, B, C, and D look like. And you can see if you compare A and C with each other, that A and C are opposites of each other, and B and D are opposites. So essentially, the magnitude of the vectors remains the same, but they're all basically related by some sort of mirror. And you can see that in the top right here. Okay, so coming back to our 3D electron diffraction, we've looked at the insights of this data. We've realized there's another polytype. What can we do now? Well, because we only are looking at such a large area, illuminating our area, the obvious thing to do is to turn to 4D STEM. So in 4D STEM now, we're looking at a much smaller area in our crystal because we're illuminating a much smaller area. I think our step size was maybe about 5 nanometers. And with this, we're getting structural information, but at higher spatial resolutions. And so the idea is maybe now we can separate form 1 and form 2 from within the same particle. So as I mentioned earlier, just working with the 4D STEM data alone is very, very difficult because of beam sensitivity limits and this basically low signal per pattern that we get. And so the pattern, the experimental pattern I've got on this slide is quite typical of maybe something that we might get from 4D STEM. If you don't have any information about the structure you're working with, this is very difficult to work with in a very meaningful way, especially because in these organic systems, the symmetry is relatively low. So that means you might get at certain orientations, very few Bragg spots seen per pattern. And this is exacerbated by a bit low symmetry. But the the 4D STEM data shows quite an interesting story. So here we have 4D STEM data taken on our spectra, Thermo Fisher spectra on a Merlin camera. We have just a 256 single chip. And you can see that there's some sort of streaking in this direction that I'm highlighting with arrows, and I've got a schematic here as to the direction of that streaking. And what we found is the direction of that streaking is parallel to the direction to the G vector that corresponds to these very strongly hydrogen bonded stacking of the layers. So we know that these layers stay relatively invariant, but that they can shift around each other. And so the streaking is an indication that these layers are actually indeed not very rigidly defined, but they are shifting around. And the SED basically reveals this presence of this. Sorry, SED, when I say SED, I do mean 4D STEM. SED is another word that we use for it, which is scanning electron diffraction. And so what's really powerful now is we can isolate the signal from one particle. So the particle in panel B, we can now isolate the diffraction signal from that particle, and we get different types of different diffraction patterns. And you can see that there's a variety of different crystallinity levels, but also of different forms. So we can see that in the blue square in panel B, that corresponds to panel C in this blue square here. And that can be indexed to form 2, whereas in the pink square, that can be indexed to form 1. Now, forming virtual dark field images from this is even more telling, because now we start seeing these domains, and we can see that these domains have very strong parallel boundaries, and those boundaries will correspond to layers in the xanthine. And you can see that I would call them striations within these layers. They look a little bit stripy. And that also, again, is an indication of even within these domains, there is some planar disorder happening. So the last thing that we did is we started to look at how we might want to model that back onto our X-ray powder diffraction data that I showed you earlier, and thinking about how our knowledge of these stacking vectors can be helpful to that model. So what we did is we created a supercell of 20 layers, and we started with the 20 layers. We just chose form 2. So those layers had a stacking pattern of ABCD, 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 ABCD until we had 20 layers. And then we randomly introduced a deviation from that stacking pattern in the form of another known stacking vector. So for example, A or B or C or D. And we essentially randomly dispersed these stacking faults in our stacking sequence of our supercell approach. But what I want to be clear about is we have very rigid translations that were allowed. So the translations between layers that were allowed were only in A, B, C, or D. And so we essentially created a stacking fault model. And you can see here that this X-ray fit is now a lot better. We reanalyzed the same data that we had before with just form 1, and the fit is substantially better. And just to compare the fits from before and after, if you see above is our form 1 with only our 1 model, and below the fit is with form 1, form 2, and this supercell stacking fault approach. And what's really neat is we also simulated the electron diffraction data just very simply kinematically with with our new supercell model. And you can now see the origin of this streaking that I was talking about in the diffraction pattern. And crucially, you can see that the center reflections that correspond to the distances between the hydrogen bonded stacked planes, those reflections are not streaked. And we wouldn't expect them to streak because we think that those remain invariant in their position. Okay, so to wrap up, 3D versus 4D STEM. So with 3D I've shown you what you can do. I mean, you have a parallel beam, you get a signal from a larger area of the crystal, you get a series of rotationally related patterns, and that allows for structure solution. So that's very important, especially if you have a completely unknown phase or structure. But what it doesn't allow you to do so well is to really see what the distribution of crystallinity within the particle that you looked at looks like. And for example, it's very hard to visualize defects. So we don't know what the twins or what the stacking faults or domains might look like and how they're distributed. Whereas with 4D STEM, you have very low signal per diffraction pattern, and you don't get any 3D information. Yeah, that's pretty true. And you often have very low symmetry crystal systems or polymorphs. So this just exacerbates the problem because you have many different orientations that could match, and you're working with very few Bragg spots. And the good side of it is that you can use your 4D dataset to create virtual dark field images, and you can really start to isolate signal from different parts of the crystal. Okay, so this is my summary. We've shown here that 3D ED can be used for small molecule structure determination from sub-micron crystals, and we can enhance our understanding of pharmaceutical microstructure observed in 3D ED, also using 4D STEM. And for example, where this is really interesting for the xanthine case is xanthine is just one planar organic molecule in a big family of planar organic molecules, some of which will be very pharmaceutically relevant. And this behavior that we have seen of planar disorder on the nanoscale, I don't believe is unique to just a xanthine structure. And I'm sure if there's any more work done on other planar organic systems, similar behaviors will be seen as well. And so understanding that behavior and that structural information will be, I hope, very helpful, and seeing at this length scale be very helpful for the next generation of drug modalities, especially as we're increasingly moving towards nanoengineering, nanoengineered drugs to deliver medicine. Okay, and with that, I will finish. Thank you very much for listening. And just some acknowledgements. I'd like to thank everybody at Cambridge who's helped me so much, especially Paul, with this work. And Roy and Duncan at GSK, Julio, who I did a lot of the X-ray powder diffraction analysis with, especially the really interesting work at the end with the stacking faults models. And then we'd just like to thank the guys at I11 for helping us, and for Rigaku for giving us access to their Chrysalis Pro software. And I'm very happy to take any questions. Thank you very much for listening.
Thank you, Helen, for that insightful presentation. It was really interesting to hear about some of your work. So we'll move on to the Q&A session. Again, this will last for about 5 to 10 minutes, depending on the number of questions that we do receive. So you do have any questions, just put them in the chat and I'll read them out for Helen to answer. So we have a question from Juyun. She said, or they say that, I was wondering, what was the total dose for your experiments? Also, did you observe any beam-induced modifications to the crystals?
So for the 4D STEM experiment, we were working with the beam current of about 2 picoamps, and that corresponds to, for our settings, a total dose of about 10 electrons per angstrom squared. That's total dose. And then it's a very similar dose for the 3D ED. And so what that means is we can't do 3D ED and then 4D STEM on the same crystal, because we do have beam damage effects essentially. So that was a big a thing that means we can't do both on the same crystal. Yeah. Oh, and sorry, you also asked about any beam-induced modifications. Not whilst we were collecting the data, but we just could not come back to the data. We couldn't come back to the same crystal afterwards and recollect. So if something happened, we weren't able to just tilt and then go again. It was kind of a one shot per crystal situation.
We have another question from Daniel. They've asked, has the interpretation of electron diffraction required dynamical diffraction methods, or is kinematical approximation enough?
So I didn't talk too much about R factors, but I mean, we dealt with our data here kinematically simply because of ease of access to a dynamical refinement software, just with what our group had access to within the timeframes. But it definitely would affect, a kinematical refinement. For those who aren't aware, a kinematical approximation assumes that your intensity is directly proportional to the square of your structure factors, which means that the assumption is that your electrons only scatter one time when they are in the crystal. But in reality, because electrons interact more strongly than X-rays, you can have multiple scattering. And so a dynamical model might potentially be needed. And I think, for example, if we had chiral compounds and the positions of the hydrogens really mattered a lot more, then dynamical refinement would be very important for that. And you can see in the statistics from our kinematical refinement that there's definitely a lot of room for improvement if you want to take on the dynamical model. So yes.
So another question, do you use common TEM grids? Will the mesh bar cause confusion in the diffraction? What is the usual rotation angle range?
Yes, I see that's from Motion. Thank you for the question. Yeah, so we use quantifoil grids, and they have essentially amorphous carbon at the bottom. And you can see in some of our diffraction, if the particle is on a grid and not on the hole, you do get some amorphous halo in the background, but it doesn't cause confusion with the diffraction because it's amorphous, and we're looking purely with crystalline stuff. And our usual rotation range is plus minus 60 in the cryos. Unfortunately, sometimes the grid bar gets in the way. Yeah, and the reason why we use quantifoil grids is because it just helps with the auto eccentric height finder, so finding the correct Z height for each crystal. That's why we use quantifoil, but we could in theory use wholly or lacy carbon as well.
We have a question from Marcus. How large was the semi-parallel probe used for the 4D STEM measurements compared to the step size? Do you see any propagation of damage through the scan?
So propagation damage through the scan, we don't see that. actually. We do see it if we go to slightly higher doses, but we just found that made the data set very difficult to work with. So we just reduced the dose until we didn't see those very obvious effects. Our convergence angle was 0.1 milliradians, and that gave us a step size, I think, of between 5 to 10 nanometers. Sorry, a probe size, yeah. So we didn't have any overlap, if that's your question. So we had a probe size of maybe, I think it was 8 nanometers, and then our step size was, sorry, the other way around. So 5 nanometers and 8 nanometers. So we were stepping, but without any overlap, and we didn't see any damage. And if we did, we just changed our acquisition conditions, basically.
While we're waiting for a few more questions, I've got a question. How many of the crystals on the grid exhibited the form 2 phase or polytype?
So this is an interesting question because there's confirmation bias here. So, I mean, I could have run more experiments, but I stopped when I found this one, and there's no. So I'm hesitant to say, oh, I found like 1 out of 10, so it's 10%. There's no way of knowing that. It's not a bulk technique. We only saw it in one crystal, and that was a crystal which was the bicrystal. So it also had form 1 attached to it as well. That's not to say that there isn't more in there, especially as we showed that we think that the form 2 exists within other crystals as well. Yeah, only one, but I'm hesitant to say that that's reflective of the bulk. Basically, I don't think that would be.
Just to follow up then, so you saw it on one crystal, but you mentioned earlier that you did the 3D ED and 4D stem on separate crystals, so you were able to do the 4D stem on the same bicrystal?
Sorry, yeah, we saw it in 3D ED on only one crystal, and then in 4D stem we saw it in domains of other crystals, if that makes sense. So we never found a pure form 2 polytype crystal, which is interesting because maybe the question is, can form 2, is that stable enough to even exist as a crystal alone by itself? Like, is that a pure polytype that even exists by itself? Will form 1 always be with it? Yeah, that's an interesting question.
So there's a few more questions in the chat. From Daniel, have you cooled the sample for 4D stem? Does it help?
We didn't cool the sample simply because the machine, like the holder, with the 3D ED, we cooled it because it was in a cryos, so it was cooled. And then with the 4D stem, we were working on a spectra, and we didn't use a cryo holder. So I haven't tried. Oh, we have tried it cooled at EPSIC, so in Harwell. Honestly, we found that it helped a bit with the beam damage, sure, but the bigger win was to change our probe the semi-convergence angle, and by making that a smaller angle, we have spots rather than discs, so are in diffraction space, and that meant that they were slightly clearer, and that made the biggest difference in terms of analyzing our 4D STEM data set. So yeah, I think we did try it, but I can't say that it helped loads.
Maybe we'll take 2 more questions. So from Heng Jiang, apologies if I'm mispronouncing your name. They've asked, I was wondering, what are the completeness levels in your 3D ED data? Could they have any impact on your results?
In terms of impact, I mean, if the completeness is too low, we sometimes are just not successful with structured solution. Our completeness was, I think for form 2, we had a completeness of 79%, and for form 1, we had a completeness of 82%, if I remember correctly, or around there. And that was enough to solve. It also depends on the symmetry of your crystal system as well, I guess, as to how much of an angular range you need. So the fact that, for example, it was orthorhombic means that maybe there's a bit more leeway than if you have a completely triclinic crystal system. But generally, if you have enough to solve, then you'll be able to solve and successfully, or maybe you don't, and you need to try other structured solution methods.
So, okay, there's a few more questions. Maybe I'll just read them all. From Martin, you mentioned that in 40 STEM, one only sees a few reflections of this low symmetry material. Did you consider using precession of the electron beam in your experiments to enhance the quality of the patterns?
Yeah, thank you for the question, Martin. That's a great point. Yes, I would love to do that, and that definitely would help. I haven't shown any results here from any precession, like scanning precession, electron diffraction experiment, but I think that would definitely have helped the cause a lot more had I done that. So that's something that we would definitely consider doing. And of course, in the group, other people are doing that. So yeah, that's a very valid point.
So from Ton, where do you think are the main areas for practically further improving the combined workflow?
Yeah, I think it's interesting. I think the 4D STEM communities and the 3D ED communities are not very well joined up currently. So the softwares that you would need to analyze your 3D ED data comes from the X-ray crystallography community primarily. Of course, there are people like Lucas Platinis who are working on 3D ED specific things. And then with 4D STEM, that's kind of another separate group, and you might be accessing that through PiperSpy or Pi4D STEM. So I think integrating the two together would definitely be make it a lot easier. The biggest thing that I see right now is just equipment in terms of, are you able to run 3D ED and 4D STEM on the same instrument? And if you are, and if you have the right detector for it, can you run those techniques on the same particle if you're able to apply a low enough dose? I think that would be unlocking the next stage and the next bit of progress.
Okay, so was it possible to separate signals from different domains in 4D STEM? Was it affected by preferred orientation of crystals? So this question comes from Maria.
Thank you, Marie, for the question. Yes, so it was possible to separate signals from different domains, but in very particular, in a way that was interpretable, it had to be from particles of particular orientations, because if you imagine you have your hydrogen bonded layers, if they're facing you face on, so you're really looking at them like this, you can no longer distinguish the depth between that in the 4D STEM data. So if you're looking at it edge on, that makes it a lot easier. So I guess that is affected, and there are data sets that were difficult to interpret if they were at a different orientation.
Okay, one final question. Do you see any chance to perform tilt series of 4D STEM data acquisition? This allows filtering out the defective part of crystals and then reconstruct 3D ED data for more accurate structure determination. This question comes from Ming Jian.
Thank you, Ming Jen, for the question. Yeah, I think it would depend why you want, when you say more accurate structure determination, yeah, I think it depends why you need it to be more accurate. I think the interesting idea would be, of course, to take at every tilt, take a full 4D STEM series and then tilt, and then take a full 4D STEM and then tilt, and take a full 4D STEM and tilt. I think, again, with the right detectors, that is possible. But the defective parts of the crystals, we've shown that that's really intrinsic to the nanostructure in this system. And I suspect with many other planar molecules as well, it will be very intrinsic. So I think it could be very difficult to really separate them in the way that you might be thinking of.
Okay, I think that's all the time we have for questions. So I think we'll wrap up today's webinar. A big thank you to Helen for sharing her expertise, and to all of you for joining us. The recording will be available later on YouTube after it's processed. We'll also send a link to the video to all registrants. So if you'd like to rewatch or share it with colleagues, keep an eye out for that. We hope you found this session valuable, and we look forward to seeing you at future webinars. Thanks again, and have a great rest of your day.
Thank you, everybody, for coming.